And for the next talk, as Andrea pointed out, there will be talk on the mouse models. So it's titled as Overview of Safety and Efficacy in Animal Models. What can we expect in humans? Given by Associate Professor Duško and Jan, and Dr. Jan.
Hi. Thank you. As Shivan mentioned, me both will present this topic. I will start by simply repeating what already Andrea said, and then Jan will explain how the toxicology and biodistribution was carried out, and then I will show you the results of this toxicology and biodistribution study. But before I start, I'm very happy to see you all again here, so good morning also from my side. So in order to bring a new therapy to children, to humans, we have to go through this very important pipeline. Before entering the clinical studies, as we are here now, we are in the midst of phase 1, phase 2 clinical trial. You have to conduct all these aspects of research where basically the animal studies are, for me personally, the most important ones, where you determine not only that the drug is functional, but it is also most important that the drug is to be shown that the drug is safe. So as already Andrea said, we have used a disease mouse model. I presented the phenotype, all the characteristics of those mouse models already in Ljubljana and also last year in Bilbao. So I won't go into the details, but as Andrea said, this mouse model exhibited the phenotype of the CTNB1 syndrome patient, meaning that it has a motor impairment, difficulties and also some cognitive impairments are observed in the mouse model, but also we observed a drop of beta-catenin protein within different, this is not this slide, sorry, and also loss of mRNA production. And as Andrea said, those animals were treated with different doses of virus by ICV injections and then subjected by different behavioral assays to determine the drug is functional. She also pointed out that we improved the anxiety-like behavior, also to some extent the motor impairment. But for me, the most insignificant and very important result is this improvement in overall gait score, meaning that we have improved this fine motor movement changes that is observed in a CTNB1 syndrome mouse model. We determined also a dose-dependent increase of the gene of interest that we are, of course, looking for. And as she said, we have also shown improvement of beta-catenin protein synthesis within different parts of the brain. And as she said, we didn't see any differences in liver and also in muscles. Although the main aim of doing efficacy studies to show that the drug is functional, we already wanted to see if the drug is safe. Therefore, we conducted a wide biochemical analysis of all liver parameters and also kidney functions. And you can clearly see here that we didn't discover any changes in liver enzymes, meaning that we gave us basically the opportunity to conclude that at this stage, drug is functional and also safe. So I will now give the word to Jan to explain how the safety and biodistribution studies were carried out.
Thank you, Duško, and good morning also from my side. And basically in this phase of project, our team jumped in and we basically started to work on a regulatory package under GLP certification. And here you can just see what such a design means. That we had two time points, and basically all these designs are discussed with the regulators, that you are getting the advices according to already established guidelines, and according also to statistical power. There is the 6 mice for sex and group. So you can basically try to multiply, and finally you will get the 96 mice per study. So this was the minimal number of animals which went to the pipeline, and basically then the age when we initiated the dosing. So it was actually the 4 weeks of age. So let's say 1 week after weaning, the animals went to stereotactic frame and the ICV injection, and then actually the monitoring of their health and body weight, food consumption, and also interim ophthalmology investigation, because the eye is basically the mirror into brain. So if something very severe neurotoxic would happen, the retina is one of the first sites where you can see the problems. And then finally, after the time period, we collected the blood, urine, and many organs, which I will show you in next slides. So what is important that actually EMA is requesting also so-called shedding analysis. So we were collecting also the urine and feces, which was also a very scientific part of the project. And this was very important also to show whether the virus is released to environment from the dosed animals. And then when we got into terminal blood collection, so under the quite simple image of the blood tube, there is a quite robust analysis which is done. So basically all samples were analyzed for the hematology, for the presence and the quantification of the white blood cells and the ratio, but also we did a full toxicology panel on the clinical biochemistry. So basically quite expanded panel, but what you are used to also for the medical doctors when you are investigated for the blood markers. And also in urine, again, in a very classical way, how also the human patients are analyzed for the presence of the markers in a urine. And what is important, that also the coagulation tests were run. So if you imagine that one mouse can give you maximally 1 milliliter of blood, it's quite hard job to basically use all samples accordingly and still to have something for molecular analysis. And then it came to, I would say, the most laborious part of job, and it was the detailed histopathological analysis. So we basically, instead of this list, I could write everything. So all organs were harvested, fixed, and analyzed by pathologists in our team. Just you know, to let you know how robust and also how responsible our approach is, that we have actually two pathologists. One pathologist is certified human pathologist, and one is certified veterinary pathologist. And these two people are working together to analyze the findings in the organs. And you can imagine that when you have 96 mice, you will always find something. It's not that 96 mice all will be healthy. But what is important and very responsible work of the pathologist is to see, is it a spontaneous lesion in the organ, or it's related to therapy application. And that's why we actually are relying on two pathologists to basically get as accurate as possible interpretation of the findings. And this is just the summary of the study. So you can see the branches of the study and overall the monitoring of body weight, which was very promising. So we got a quite good in life readouts that nothing really bad is happening to animals. But then the final analysis came, and now I would like to pass the mic back to Duško to sum up the results.
Thanks, Jan. So as Jan nicely presented all the pipeline, you can clearly see here that a lot was done. So we performed a 28 days long biodistribution study, 90 days long tox and biodistribution study, and additional 180 days long biodistribution study. And here are the results of the normal development of the animals that were put to this 90 days long tox and BDS study. And you can clearly see here that we didn't observe any changes in body weight in the treated animals compared to the vehicle-only treated groups. For the first panel, I'm showing you the survival rate of the animals that were included in this 90 days long tox BDS study. And you can clearly see here that we have 100% survival rate for female animals, whereas we observed 2 deaths of the animals that were put in the mid-dose group. Of course, we were at that stage very, very worried, but we did a thorough histopathology assessment of these 2 animals, and they exhibit signs that were then based on the wide literature search and also consulting with worldwide experts regarding veterinary pathology. We came to the conclusion that this could be due to the back strain of the animals, and also due to the fact that both male mice originated from the same litter. And we basically determined, or we conducted, as I showed you on the previous slide, the same dosing regimen also for 180 days long biodistribution study and also for the 28 days long biodistribution study, where we didn't discover any changes in mortality of the animals. And we also only observed 2 deaths of the animals in the mid-dose, now in the higher dosage treated animals. As Jan pointed out, a vast clinical chemistry and also biochemical analyses were performed on blood and also on urine. And for us, we were quite happy to see that we didn't discover any changes in the clinical chemistry profile, none in the coagulation parameters, and also we didn't discover any changes in the kidney and also liver functions. So we can conclude here that we didn't observe any changes on the blood and also on the urine profile. And based on the clinical scoring that was mentioned by Jan, we didn't also discover any clinically relevant changes related to the treatment. As Jan pointed nicely out, the most important thing for us was, of course, to test a whole body histopathology assessment. And we were very focused on, of course, on the brain. And here I'm showing you here on the heat maps a scoring of each animals of the brain region where we tested, or the pathology tested, or looked for inflammation, edema, hemorrhage, scarring. And here I'm just showing a representative images of vehicle and also high-dose treated animals of the brain morphology. And we didn't discover any changes in brain morphology in high dose, mid dose, low dose compared to the vehicles, because you can clearly see here that the colors are pretty much the same. And here's just the summary, because I don't have enough time to go over every specific parameters also for this type of studies. These are just the clinical scoring for all the animals in the 28 days long biodistribution study and also normal development. No body weight change was observed, and also within the 180 days biodistribution study, again, no clinical changes observed in any animals, and also normal development in the male and also in the female groups was seen. In Australia, also they did a non-GLP long-term toxicity study where they treated the animals, the wild-type animals, on the same delivery route as we did in GLP-certified tox study. They infused the animals with super high dose and high dose, and then, of course, the animals were subjected to further observation for additional 8 months. And we didn't discover any changes in the development, body mass, and then at the end, again, all the brain tissue, liver tissue, kidney tissue were observed, where we were quite happy to see that we didn't discover any changes in the thickness of different brain areas. So concluding that we can determine that we didn't observe any long-term toxic effects. At the end, before initiating or before submitting all the relevant papers to the regulators, we also conducted a toxicology and also biodistribution study on non-human primates using Macaca fascicularis monkeys, which is a pretty standard model. And here we already use a GMP-like tox batch called Urbagen. You all know why Urbagen, right? And we have used animals where we tested first 2 delivery routes, where we injected the drug of choice via intracisterna magna injections. And then we also did bioventricular ICV injections containing low dose and also high dose. And these animals were also then subjected to further clinical observation for 90 days, where at the end we analyzed all the tissue for vector and also transgene expression via modified qPCR. And of course, blood panel tests were done to confirm the safety of the drug, and of course, final terminal histopathology assessment. You can clearly see here that in all tested groups, animal developed normally, and the clinician also didn't discover any changes in reflex presence, and to our satisfaction, we didn't see any negative effect on the liver parameters and also on the coagulation status of the blood of the animals, confirming that there was lack of liver toxicity and lack of potential TMA occurrence in the animals. As Lainšček also pointed out, AAVs can cause some side effects in the living organisms, where the most detrimental, the most worrisome are liver toxicity and also dorsal root ganglion toxicity. And then by doing the final terminal histopathology assessment of those mentioned tissues, we were quite happy to see that we didn't discover any DRG and nor liver toxicity. And we were really happy when we got back those results. At the end, as I mentioned, all the animals were of course euthanized, and the tissue were then analyzed by a ddPCR to determine the copy number of the vector and also to determine the copy number of the transgene expression, meaning our human CTNB1 gene. And you can clearly see here that we have a wide organ distribution of the vector within different parts of the brain and also different parts of the organism. And it is also observed same for the transgene expression. And also we observed a lower liver expression of the transgene, meaning that the microRNA binding approach that was presented by Andrea is working. And with that, I would like to thank all the people that basically helped us achieve to get to this point where are we today. And I would like to thank you for your attention.